Okay, right. Moving right along, chapter 11, and this is on cytotoxic T lymphocytes. Okay, so as you know, viruses are obligate intracellular microbes. Um, Killing the infected cell, the entire cell, will cripple the ability of the virus to survive in the host. And this is done through CD8 positive T cells or cytotoxic T lymphocytes. So cytotoxic T lymphocytes eliminate intracellular microbes by killing infected cells through mechanisms that include perforin granzyme and fast ligand. So the basis of this immunologic response starts in the same way of every response that we've reviewed throughout the year, that you get the microbe, in this case, for example, a virus, which is still engulfed by a dendritic cell, where then in the lymph node, it encounters a T cell, but then through different co-stimulation and cytokines, this will become a CD8 positive or cytotoxic T lymphocytes, which will then migrate to the site of infection instead of a CD4 positive T cell. That being said, CD4 positive T cells are still a part of this larger broad pathway because they will co-stimulate those cytotoxic T lymphocytes, and they will produce cytokines that stimulate this very specific differentiation, and then they enhance expression of the co-stimulators that we'll need to activate. So here is just a nice drawing of exactly that. You have your antigen presenting cell, such as your dendritic cell, and this would be in a lymph node. It has already engulfed the virus, and then through help with a CD4 positive T cell, certain cytokines will signal to the naive CD8 T cell, along with certain co-stimulators that will then cause this differentiation into both memory CD8 positive T cells as well as cytotoxic T lymphocytes. So specifically, some co-stimulation that can occur is through CD40 and CD40 ligand, which we have heard about before, in addition to that type 2 MHC. But it is through, when it recognizes CD8 positive T cell, this signals through MHC1. And then you can also get a feedback. Well, once this is stimulated in the correct way, the dendritic cell has made a determination of what the milieu should be. It can also secrete cytokines that will cause the same expression of CTLs. Okay. So differentiation and survival of cytotoxic T lymphocytes, no different than any others. IL-2, again, promotes proliferation and differentiation. IL-12 and interferon will promote differentiation from a naive CD8 cell to a effector CD8 cell. IL-15 is important for memory CD8 survival. T-bet, immunodermin, and BLIMP1 are transcription factors, and then cytokines like typical cytokines will signal through the JAK-STAT pathway. So these are not that different or unique. One thing I would probably take out of all of this, one, IL-2, you should be associating with T cell survival at this point, and IL-12 interferon towards this CD8 axis. And then hopefully by this point, you're starting to recognize that cytokines typically signal through the JAK-STAT pathway. Okay, there is chronic viral infections and cancers that can exhaust cytotoxic T lymphocytes. This is from repeated stimulation that results in decrease in proliferation, a reduced production of interferon, and then CD8 cannot clear the infection. So just like in the case of a granuloma, where you actually, your macrophage cannot finish clearing the infection, there is a chronic viral exhaustion that can happen in cytotoxic T lymphocytes at the same time. So hopefully if everything goes right, you get that naive T cell that will correctly differentiate into a cytotoxic T lymphocyte, travels to the site of infection, and then it will recognize these virus-infected cells through this MHC1, and it will signal for this cell to be killed, and thus the viral infection will go away. There is a situation, though, when you can exhaust these CD8, get inhibition, and then you can have this live infected cell that persists in the body. Okay, so the two pathways of effector function are through perforin granzyme and Fas ligand. Okay, so in the perforin granzyme pathway, what happens is the cytotoxic T lymphocyte will bind to the target cell by the antigen receptor, co-receptor, and adhesion molecules, and then it initiates cell death by apoptosis. So it's a controlled cell death instead of the cell just bursting open and releasing the relatively inflammatory interior of the cell. So then granules will contain perforin and granzyme. Perforin actually will be released and drills holes into the membrane of the target cell after that stable adhesion between the cytotoxic T lymphocyte and the target cell happens. The two will closely approximate. These granules are released. Perforin will drill holes in membrane of the target cell. And then granzyme enters and it cleaves caspases that lead to the death cascade. There is something called cerglycan that will assemble granzyme perforin complex, and it's probably the only other piece of this that you really need to know. Perforin granzyme in general, I probably just remember as a system. This perforin granzyme system is delivered by cytotoxic T lymphocytes, and it leads to this organized death cascade instead of this an inflammatory type of cell death. So here is a nice demonstration of the stable adhesion that occurs between these two cells. So you get the cytotoxic T lymphocytes, you get your target cell. This is presumably virally infected. And then you get this adhesion between the MHC that's displaying a piece of the virus, the T cell receptor, you have the co-receptor with CD8, and then you have this integrin ligand that will hold this stable and close. Once that happens, then it can release these granules, and then it will target this. The cytotoxic lymphocyte can detach, and the target cell will die. Here is actually a very nice picture of this interaction that happens where you get this synapsis, which we've discussed before, between the cytotoxic lymphocyte and the target cell. And then you can see here the synapse again. It's nice and big. It demonstrates how many different receptors are actually at play that are holding these together. So this is a very controlled event, that the perforin is just not out in the periphery perforating holes in all kinds of cells. It's a very close, stable adhesion that will allow this to happen. You can see that up here in the right. The other cell function for cytotoxic T lymphocytes is through this Fas ligand. So the cytotoxic T lymphocyte will express Fas ligand, which will bind Fas on many different cells, and then this interaction will directly activate the caspase cascade without granzyme entering the cell. So here is, again, this perforin granzyme system. You get this close, stable interaction with lots of different cells. You release, and this perforin granzyme will start the apoptotic event. And this one, it is just a direct Fas ligand mediated killing without the T cell receptor MHC association. Oh, maybe it does. I take that back. I think this is supposed to be MHC, and this is cytotoxic T lymphocyte, which will then lead to a Fas ligand interaction. So I guess it samples first to make sure it is make sure it is infected through the T cell receptor with the MHC class 1. And then there will be an interaction between Fas ligand on the cytotoxic T lymphocyte and Fas on the target cell, and that will initiate apoptosis. Okay, and that's it. Very, very quick today. Oops.